So here I'll be briefly talking about two very important things. One is MetaMine and also the advanced biomarker discovery pipeline that we have in MSOne. So before we go into MetaMine, let me just quickly talk to you about a small study that happened, actually a rather big study that happened a few years ago. And this one is about pancreatic cancer. Now, pancreatic cancer is, of course, a very serious condition, and it is very important when you have such conditions that you identify biomarkers associated with these diseases. But oftentimes, the authors noticed a particular issue that many different authors from different parts of the world they perform metabolomic studies and they publish their results. But there is not much consensus between the different studies. Each author might report some biomarkers, but that might not be common with others. So after they noticed this issue, they decided to perform this meta-analysis. So what they did is they combined results from almost 24 published studies to filter out false positive biomarkers and try to create a consensus robust biomarker panel that can be used to predict pancreatic cancer. So the overarching goal of this particular paper is to increase biomarker robustness through cross-study evidence. Now, without going too much into the details of this particular study, what they ended up with is creating a consensus metabolite panel of almost 10 different of these metabolites. And this does obey the physiology and the behavior of pancreatic cancer. And this was created through a consensus study between different publications. So this highlights how public data can be extremely powerful and you can perform different studies even without you having to do experiment yourselves. So that is the power of public data analysis. So how can we perform public data analysis? There is a very large growing body of metabolomics research in the public domain itself. And at present, there are more than 5,000 datasets available across many different public repositories, and these cover more than petabytes of data. So this gives you the opportunity to perform many different kinds of research projects without doing any experiments whatsoever. And another important advantage is that some of these datasets might be from a few years ago, and the authors of those times probably did not have access to the advanced algorithms that we have today, like the ones that we present in MS1. Also, importantly, MS1 is a very important tool for public data mining because, first of all, it has a vendor agnostic design. And on top of that, it has very powerful deep learning algorithms that extract meaningful information from this raw LCMS data. And there is a lot of very transparent and easy to use technique, options to visualize the raw data, to perform statistical analysis, etc. So MS1 can be a very good companion for this purpose of public data mining. So because we identified this, the opportunity to do this, we decided to build MetaMine. And MetaMine is basically a repository of public domain metabolomics datasets that has been processed using the AI-powered algorithms of MS1. So where do you get these kind of studies? There are a few repositories, for example, Metabolomics Workbench, Metabolites, etc., which are repositories where authors submit their raw data sets, and from them, all of us can download it and process them as required. So we have already taken the effort of downloading the data from there, processing it using MS1, and we have curated them for your use. And this is MetaMine is not something that is new to you. You have always been using this in this particular tutorial, and we will see that in a minute. Prior to that, we'll give you one more example of how public data mining can be utilized. So this is a study that was done by our team recently. We noticed that oftentimes we use blanket fold change cutoffs and p-value cutoffs to find significant metabolites in our data. So we thought instead of using a blanket fold change, what if we evaluate the inter-individual variability of metabolites in the healthy population to make a more informed decision of how much fold change can be expected in a particular metabolite? I'll repeat. Most metabolites have some degree of inherent variation, and that is not uniform for all metabolites. So we thought, instead of using a blanket fold change cutoff, what if we could identify the inherent variability of these different metabolites? So in order to study this, what we did is we took 14 blood plasma datasets from the public domain and we combined them together and we created a pseudo cohort of almost 800 individuals, and this covered almost 300 different metabolites. So we got access to almost 800 individuals and almost 300 metabolites without having to do any experiments ourselves. And to quickly show some of the results, we did evaluate some of the variabilities of these different metabolites. And we noticed that, for example, clinical biomarkers are very tightly controlled, for example, cholesterol, whereas some other molecules like bile acids are very highly vary metabolites. And maybe if you use a normal fold change cutoff of 1.5 or 2, that might not actually reflect the kind of fold change you can expect in bile acids. Now, let us quickly take a look at what MetaMine is. Just a second. Yes. So as usual, I'll click Get Started once I log in, and on the top left we can see the MetaMine button. So now you have this page that says MetaMine datasets. And as you can see, at the moment, there are 100 projects on this repository, and this is ever growing. We will keep on adding more and more datasets to this repository. We have almost 8,000 files in this. So that's almost 8,500 injections from all over the planet that has currently in this repository. And we have almost 32,000 different annotated features across all of this. And if you scroll down, we can see the tabular display of this repository. We have the first column is repository ID. So you can see some pattern here. Some of these start with MTBLS something, and some of them start with ST00. So this depends on the repository from which we got these datasets. If it starts with MTBLS, that means it came from the metabolites repository, and ST00 means it came from the metabolomics workbench repository. We also have the project name, the sample size, species, etc. So species, we can see that it is dominated by Homo sapiens because there is a lot of interest in human studies. But at the same time, we have covered many different species, whether it's mice, bacterium, plants, etc. So if you scroll down, we can see even plants like Arabidopsis. So there is a really good diversity in the kind of species we have covered in this repository. And even in the organism part also we have a lot of diversity. We can see feces, blood serum, urine, there is bacterial cultures, there is tissue of different human body parts, etc. And we can also see the column type. The column type can be either reverse faced or normal face, etc. And we can even see the kind of instrument it was measured on. It can be either an orbit trap or a triple TOF, etc. And if I open one of them, let's open the same one that we have seen in this tutorial, which is the Guillain-Barre dataset, MTBLS 10392, which my colleagues already used as an example. If I open it, we can see, the ID of the study, some details about it, the species, organism part, or etc. And as you have already seen in this tutorial, you can always load any of these datasets and use them for your studies. So currently we were working on the positive ion mode, and we'll continue on that for one particular very important part of MSOne, which is the deep analytics pipeline. So in order to see it, I'll go to the left side pane and click on Metastat. So we have already introduced Metastat in the previous session where we talked about, the pre-treatment parameters, the exploratory analysis, volcano plots, etc. And at the very end of it, we have the deep analytics pipeline. So let us quickly, talk what exactly this deep analytics pipeline is about. You know, in this present day, machine learning is a very strong tool that we can use for many different methods in daily life as well as in our research. And oftentimes it is difficult to decide what kind of a machine learning approach should you use for your research question. And it's also difficult to implement some of these. So we have automated some of this into a deep analytics pipeline so that you can use it directly without having to spend too much time into the nitty-gritties of it or to build the entire thing yourself. So when you visit this deep analytics tab, you will initially be given some options, the blank subtraction, normalization, all of those options will be presented before you. And once you submit it, there is some processing time, and after that you will get this PDF. In this case, we have already generated the PDF for you. And let us quickly see what is in this deep analytics biomarker discovery report. On the first page is something that we have seen a while ago, which is the results of the significant metabolites through a volcano plot. So this is purely based on fold change and p-value, which is fairly intuitive. We can see which are the upregulated and downregulated metabolites. And if I scroll further down, we can see these are some of the significant metabolites in the form of a heat map. So instead of you having to do this manually, we have automated that process for you. We can see significant metabolites, and we can also visualize that in the heat map. And if I scroll further down, we have the first unsupervised method, which is the PCA. PCA is something that is familiar to all of us. It is basically a dimensionality reduction method. When you have multidimensional data like metabolomics, it compresses that into maybe 2 or 3 dimensions so that you can easily visualize it. In this case, for example, I can see that there is one outlier here. So you can interpret PCA as you wish. And if I scroll further down, we have the PLSDA plot. Now, PLSDA plot is a supervised method. It looks at the label of the sample and tries to segregate them as much as possible. In this particular example, there are two classes, healthy and Guillain-Barré syndrome. So it tries to segregate the healthy and Guillain-Barré classes as far as possible and tells you which are the metabolites that contribute to it. Now, I'll skip directly on to something called AutoML. So what exactly is AutoML? AutoML is basically a collection of ML algorithms where you do not have to worry about choosing the right ML algorithm. So basically what it does is there are multiple options. Maybe there are 10 different ML algorithms. It uses each one to find what is the best way to segregate the disease and the control population and presents this together into a simple black box kind of tool where you do not have to worry about which ML algorithm is used or what is happening behind the scenes. It packs it together into a clean method where it uses help from multiple algorithms to give you the result. So how does it do it? It first takes maybe 80% of the samples as a training data set and it studies it first and it validates its method on a test data set. So when you do that, it gives you the accuracy, the sensitivity, specificity, etc. So this is to sort of validate the quality of the predictions. And if you scroll down, it will tell you something called a SHAP value. The SHAP value is basically the contribution of each metabolite to classify a sample as disease or control. It tells you how much each metabolite contributes into segregating these two as disease or control. And this can be seen, for example, in something called a waterfall plot. So this is something called a waterfall plot for a single sample. So this represents one individual and we can see multiple metabolites. So this basically shows how much each metabolite is contributing to push that sample into the disease class. So if this individual, so in this particular case, this individual is in the disease class, it tells that, okay, this metabolite slightly pushes it into the disease class and the dipeptide at the top, it pushes it most into the disease class. So once again, the SHAP value tells how much each metabolite pushes one individual into this category or that category. So that and this as a list is finally given on the last page. So to quickly summarize this deep analytics pipeline of MS1 uses multiple different methods, which includes simple methods like volcano plots and even more complicated methods like AutoML, to quickly give you an overview and uh, enable you to find biomarkers in your dataset with little effort and without you having to spend too much time on the actual, the difficult ML parts of it. So that concludes this tutorial on MS1.